Dear student, I'm Dr. Nidhi Sharma, Associate Professor, Zoology, p.g.govt.college, Jhalawar, Rajasthan. In this part of the muscular system, I shall tell you something about the sliding filament theory. Now, a sarcomere is a functional unit of skeletal muscle. When a skeletal muscle contracts, sarcomeres shorten. This is described by the sliding filament theory. Now, in this case, it is being depicted by this diagram. Here, you can see the cross bridges of the myosin filament of the thick filament depicted by the orange color and the actin filament which have got the brown and yellow globules. The sarcomeres shorten, become thick and thin filaments because thick and thin filaments slide past one another. Thin filaments move towards the center and the myosin binding sites are present on the actin protein. The myosin cross bridges bind to the thin filament. Here you can see a photograph of the sliding filament mechanism. In this photograph, you can see that in the middle photograph, the H zone has been reduced, and in the third photograph, you can see the zone has completely disappeared and the muscle has shortened in size. In this, in the ray diagrams, you can see that the two sarcomeres have been shown. Right from one side, you can see the zigzag blue colored Z disc to the next Z disc, which is zigzag and blue in color. This is an elongated muscle which is not contracted. Here you can see the prominent H zone, the I band and the A band. In the middle figure, you can see that the H zone has reduced considerably and the I band has also shortened. This is a partially contracted muscle. In the last figure, you can see that maximally contracted muscle wherein the H zone, which is shown by the green shaded region in the relaxed muscle and partially contracted muscle, has completely disappeared and the muscle has become short in length. The sliding of actin on myosin, which are thick filaments on thin filaments, can be broken down into a four-step process which will be shown in the next slide. Now, I'll tell you something about the physiology of skeletal muscle contraction. This is done by neurotransmitter release In this case, the neurotransmitter is acetylcholine molecule. When the motor impulse is initiated in the brain, it travels through the brain and spinal cord to a motor nerve ending. Motor nerve endings, that is axons, depolarize. In a diagram, in the next slides, I will show you the depolarization and repolarization of the muscle. Now, the calcium enters the axonal endings. Calcium ions cause the release of acetylcholine into the neuromuscular junction, which is the tip, synaptic cleft. Here in the above diagram, you can see the axon fiber, which is depicted in yellow, and the small blue colored synaptic vesicles and the synaptic knob along with the calcium ions, and you can also see a few mitochondria. This structure of the synaptic knob has been elaborated in this diagram. Here you can see that the nerve impulse is carries and the synaptic vesicles carry the neurotransmitters. Now the synaptic vesicles release the neurotransmitter near the axon membrane. The neurotransmitter is depicted by blue circles, and in the synaptic cleft, the polarization and the depolarization of the nerve membrane takes place. You can see the positive sign inside the synaptic cleft and negative side on the opposite side. Then again you can see positive ions and then negative ions outside and positive ions inside the cleft. In the next slide, I will tell you about the depolarization and the repolarization of the membrane. Acetylcholine binds receptors on motor end plate, depolarizes skeletal muscle fibers, and gives reverse charge to the membrane. As impulse travels through transverse tubules to reach all of the muscle fibers, muscle depolarization causes release of calcium from the sarcoplasmic reticulum into the sarcoplasm. Here again in this diagram, which we have already studied, relationship of the sarcoplasmic reticulum and T tubules to myofibrils of skeletal muscle has been shown. Now, there is something called the power stroke. What happens in a power stroke? What is the physiology involved in the power stroke of a skeletal muscle? Here, calcium binds troponin, which is attached to tropomyosin. This moves tropomyosin from the myosin binding sites on actin. Myosin cross bridges bind to actin. These cross bridges are globular head of the myosin. Here ATP is hydrolyzed to supply energy by ADP plus inorganic phosphate. Then actin is pulled inward towards the center of the sarcomere with the help of these cross bridges or heads, which is known as the power stroke. Sarcomeres shorten as muscle contracts. Here in this diagram, the sliding filament mechanism has been detected. It has been depicted. Now you can see the actin filament depicted by light orange color and the myosin filament by the purple blue color. The myosin filament has a globular head and a neck which touches the actin filament and forms the cross bridges. These cross bridges pull the actin filament inside, and in this way muscle contraction takes place. With exposure of the myosin binding sites on actin, the thin filament, in the presence of calcium ions and ATP, the thick and thin filaments slide on one another and the sarcomere is shortened. Here in this diagram, the cross bridge attachment to the actin fiber is clearly seen. In the first step, with the help of hydrolysis of ATP, the myosin heads or cross bridges hydrolyze, that is, break up ATP in the presence of water and become reoriented and are energized. In the second step of attachment, myosin heads, the orange colored globules, bind to actin, forming cross bridges. Now, this diagram depicts the cross bridge cycle. In the first case, as can be seen, myosin head attaches to the actin myofilament, forming a cross bridge. In the second stage, inorganic phosphate P1 is generated in the previous contraction cycle, which has been generated in the previous contraction cycle, is released. This initiates the power stroke. The myosin head pivots and bends as it pulls on the actin filament, sliding it towards the M line. Then ADP is released. In the third stage of this cycle, as new ATP attaches to the myosin head, the link between myosin and actin weakens and the cross bridge detaches. Then, in the next cycle, as ATP is split into ADP and P phosphate, inorganic phosphate, the myosin head is energized. And once again, it is cocked into the high energy conformation. Here in this figure, the globule is purple globule is the calcium ion. Now you can see how myosin heads, which is the orange, myosin is the orange filament with globular heads, hydrolyze ATP and become reoriented and energized. Here, the myosin heads have been attached to the actin along with the calcium ion, and in this case, the myosin cross bridges rotate towards center of sarcomere, which is the power stroke, and pulls the actin fiber towards itself and shortens the muscle for contraction. In this diagram, as myosin heads bind at ATP, the cross bridges detach from actin. In the next part, I shall tell you about the excitation contraction coupling.